Hey guys, Mr. Hyatt here. This is the AP Biology Chapter 24 lecture on the origin of species. So we start out looking at the Galapagos, of course, and what is really powerful about the Galapagos is they're an island where we found lots of life that doesn't exist anywhere else on Earth. So what is it about this island that caused things to get so different so fast? A couple of kind of basic things here. Speciation, we should know that by now. That's a process by which one species splits into many. Microevolution is going to be small scale changes within a population. Macro is going to be large scale changes above the species level. So huge evolution, basically. Micro is always small, macro is always big. There's an animation here. You guys know I can't play these animations very well in these flips, but make sure you check Canvas to get the animation here. It's a good summary of macroevolution. So if we're talking about how we create new species, not how we create new species, but how new species are created, we need to have a common definition of what a species is. Unfortunately, there are multiple definitions of what a species is. We use, here in AP Bio, we use the biological species concept. Can you make babies that can make babies? If so, you're the same species. If not, you're not the same species. But there are some issues with that. We've got diversity within a species, and we've got some similarity between different species. So what about things that can kind of mate and make babies? Well, they're not fertile offspring. So what's up with the hybrid? What's up with a mule? Where does it fit in the grand scheme of things? Where do things like ligers and tigons fit? If I flip back, to this one, you'll see that we compare morphology. That's like shape, physiology. That's like how we function. So anatomy is what the structures are. Physiology is how you use them. So morphology is what you look like. Physiology is how you move, how you do things. Biochemistry, that's going to be how you process things, and DNA sequences, we know what that's all about. So when we look at morphology and physiology, there are definitely some morphological differences between these six folks. Their skin tone is a little bit different. Their hair texture and hair color and uh, their eye color and they just have lots of diversity. And then here these two look really similar, but they're a different species. So you can see why the different types of the different systems are tricky. You'll see there's a reason that we use morphological shapes. Think about the fossil record, but you'll see that later. Another video, make sure you watch that. Again, apologize, I can't play those animations and videos. But this whole thing is going to be based around reproductive isolation. If you're reproductively isolated, even if you can meet and attempt to mate, you can't make fertile offspring. So that reproductive isolation is going to happen one of two ways. We've got prezygotic barriers that are going to happen before the zygote forms, and then we've got postzygotic barriers that are going to happen after the zygote forms. Here are some examples. We're going to go through a lot of these. I've deleted some of the pictures on the presentation just to cut down on our number of overall slides, but you can see there are a lot of prezygotic barriers and there are 3 postzygotic barriers. So before I get too deep into it, prezygotic barriers are going to block fertilization. Could be keeping the species from completing the act of mating. That would be the second one. Could be keeping the species from attempting to mate, so keeping them separated. And then hindering fertilization if the mating is successful. So again, prezygotic is prezygote. 3 types of prezygotic barriers we're going to look at first: habitat isolation, temporal isolation, and behavioral. So habitat isolation is really easy. If you're not in the same place at the same time, you can't make babies. So maybe one population lives on an island in the Atlantic, one population lives on an island in the Pacific. Those 2 gene pools are isolated, so we're going to accumulate our own sets of mutations, and we're going to come up with our own Atlantic version of the species and our own Pacific version of the species. Temporal isolation is going to be temporal means time, so they're going to be isolated by differences in time. Maybe we've got species that breed at different times of the day. Maybe we've got one type of raccoon is active during the day, one type of raccoon is active during the night. Since they're not active, they're sleeping at opposite times, they're temporally isolated. More realistic examples would be things like cicadas that come out every 7 years and things along those lines. Different bears are hibernating in the winter and they're active during the winter. You know, a polar bear is going to thrive during the winter and a grizzly bear is going to hibernate during the winter. So that's another example of temporal isolation. Behavioral isolation is going to be mating dances, basically. Courtship rituals and other behaviors unique to that species are effective barriers because there's a video here that shows these types of birds. Look how blue their feet are. You'll see them when they start to dance. But a blue-footed booby is going to do one type of mating dance, and there are other colored boobies. Those are types of birds. That's what this species is called. There are other color-footed boobies that do different courtship rituals. So if you don't do the right mating dance, I'm not going to mate with you, is the idea. I've got a couple of examples here. I've got these birds, I've got albatrosses, and I've got giraffes. So all different types of things. You have to act right to be chosen as a mate, essentially. Next two, mechanical isolation and gametic isolation. You'll see these two really play off of each other. Notice that this is, here we had no mating attempt, no mating attempt, no mating attempt. Okay, so these kind of get chunked apart. I wouldn't worry about how they're chunked apart. I think I would just try to understand them individually. So mechanical isolation is the parts got to fit together. I've talked about snails several times throughout the course of this year. There are some snails that go, their shells wind clockwise, some that wind counterclockwise. The parts don't match up, so they can't mate. They can't make babies. And then gametic isolation is where the sperm of one species may not be able to fertilize the eggs of another species. Maybe they're chemically incompatible. Their cell membrane structure is so different that the sperm can't get into the egg. Something along those lines. Moving on to postzygotic barriers. Those are going to be sperm and egg meet. Egg is fertilized. Then something happens. We've got three of them right there: reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. So viability is livability. So basically a hybrid is less fit. It's going to be weaker. It's going to be slower. It's not going to be able to survive somehow. It's going to be less fit than is a true breeding member of the species. Or it could be reduced hybrid fertility. So the hybrids are healthy, like this mule right here. The hybrid is healthy, but it can't mate. So the number of chromosomes are odd. That's what tends to be the case in hybrids. We end up with an odd number of chromosomes. So when this mule attempts to mate, it tries to go through meiosis, and the horse's body recognizes, hey, something is wrong here. There's an odd number of chromosomes. We can't do independent assortment. We can't do meiosis. So then, sorry, I thought I had already had that on another slide. So then, thirdly, is hybrid breakdown, where the first generation hybrid is fertile, but when they mate, the offspring can't make it, basically. Sometimes they can make it, but they're not sterile. We see this a lot in crops, where we can have a second generation, but we just can't get a third generation. There are definitely some limitations. I referenced this earlier. We can't apply it to fossils. It's tough to apply to asexual organisms, which includes just a small group of organisms known as prokaryotes. But it works for things that are alive today that reproduce sexually. It's really good for that. And it works really well for intro-level biology. Once you start getting really deep, you have to think about some other definitions of species. So gene flow is huge because, like it says here, biological species emphasizes the absence of gene flow. But that can be tough because we can do things like roller bears and ligers and tigons, and there are some examples of some pictures of a grizzly and a polar that crossbred and made a hybrid roller bear. And you've probably seen pictures of ligers and tigons and things like that. So there is gene flow between species because we have interspecific breeding. Now, this organism is not going to be able to pass on those mutations, or maybe the white hair color is a really great adaptation for the grizzly bear, but it doesn't matter. It's going to have hybrid infertility. So those other definitions of species that I referenced, morphological species says if you look alike, you're the same. Does work on sexual and asexual species, but if you look alike, that's going to be a subjective thing. How close are you looking? Does brown hair and brown eyes count? Blonde hair, brown eyes is a separate species? I don't know. Ecological species concept is basically, in terms of its niche, really emphasizes the role of disruptive selection because middles are difficult to understand. If you're this or that, if you're really fat or really skinny, makes sense. You have a different niche. But if you're in the middle, then you probably don't have a niche. So think about some of the pictures that we saw when we looked at ecology that showed niche partitioning and how birds share trees and things along those lines. But like it says here on the screen, there are a lot of different definitions of species, and it really depends on what you're trying to do with it. So for our purposes, we mostly say make babies that can make babies. So speciation can take place with or without geographic separation. We're going to look at a couple of these, allopatric and sympatric. You don't have to be geographically isolated to become reproductively isolated. An example here would be cichlids, or cichlids. They're kind of either way is okay to say. Allopatric speciation is going to be other country, other homeland speciation. So that's when you become isolated. Flightless cormorant, I think is how you say that, of the Galápagos, likely originated from a flying species, and then some flew out, and then they accumulated mutations over the course of a couple of million years and became a flightless bird. So in that case, that flightless bird would never be able to impact the homeland species because they're separated. One's on an island, one's on the homeland. Sympatric speciation, they happen in the same pond. They happen in the same place. Notice that on this picture there's a break there. In this case, the waters rise, so we get a connection of these two ponds, for lack of a better term. So how does allopatric speciation work? Well, a canyon could create a barrier, obviously not for birds, coyotes, or pollen or something like that. So your barrier is going to be dependent on what your species is. Sometimes a river can flood, and that can cause allopatric speciation. Sometimes it doesn't.